Good morning, everyone. Thank you so much to the organizers for having me here. I'm going to tell you a pretty different story, I guess, but that also pertains to prefrontal cortex in a completely different aspect that has to do with numbers of neurons. So Nenad was just telling you about differences between mouse and macaque and human cortices, and one of the key differences, I would say, of course, that's my bias, that the most striking difference that you see across these species is that we have 1,000 times as many neurons in the cortex as mice do. What are the consequences of having such different numbers of neurons? I'd like to show you some data, but I'd like to first share with you a piece of my mind about why do we care about cerebral cortex to begin with, and prefrontal cortex in particular, and that has to do with what does a cortex actually do. I teach neuroanatomy, and one of the things that I love to point out to my students is that first, technically, nobody needs a cerebral cortex. And what I mean by that, and there's beautiful work, by the way, showing that when you have genetic disconnection, a genetically caused disconnection between the thalamus and the cortex, between the rest of the brain and the cortex, you still have animals that can behave. My point is that the parts of the brain that actually operate the body are the ones located in the brainstem. That's where you find all the loops, the effector sensory loops that produce and regulate behavior and literally keep your body alive. And that includes, by the way, the hypothalamus, which is this front part of the brain here. The cerebral cortex is this part of the brain that saddles the rest of the brain. And I really mean saddles, and I'll show you why in a minute. And what connects the two things together is the thalamus, which relays pretty much every single piece of input that the other parts of the brain receive. Notice that this means that we typically describe pathways in terms of starting somewhere and ending in the cortex, but that's really a misnomer, because these are pathways that branch out and end in many other structures at the same time, as they also form collaterals to the thalamus, which then go on to relay those signals to different parts of the cerebral cortex. And then the real magic happens once you have now the possibility of having something else going on in this cerebral cortex that could not happen in other parts of the brain, which is this possibility of associative processing, thanks to these the distributed connectivity followed by the associative connectivity that you have in the cortex, and in particular in prefrontal cortex, which is the part that is by functional definition, not connected to actions nor to sensations. So this is really that's one way of defining premotor associative cortex. It's cortex that's neither motor or visceral, and also not sensory. Okay, so that's the cerebral cortex. It's this part of the brain that is capable of creating new associations that weren't there before, that other parts of the brain couldn't do. What do you gain with that? I'll say in one word, you gain flexibility. Your behavior becomes flexible. And here's a whole other story, but I need it for the purposes of my own work. I needed to come up with an operational working definition of intelligence, and that is what I use. It's intelligence, it's behavioral flexibility. It's the possibility of acting differently, of doing things differently over time based on experience, of course, but really in a way that maximizes your future states. That is what we gain with flexibility. Now, the flexibility in this case arises once these all this associative connectivity in the cortex is capable of acting back on the other parts of the brain and modulating their function. So flexibility now not only modifies actions, but makes complexity possible. So I like to think of complexity as one more way in which flexibility presents in adding to behavior. So behavior with a cerebral cortex becomes flexible, becomes complex, and then it starts snowballing into the different forms of flexibility and complexity. For instance, you gain not only adaptive behavior, but adaptive behavior is something that every self-organized system have. I think that what we really, really gain with a cerebral cortex and a prefrontal cortex in particular is the possibility of acting in anticipation of what comes next. And that is something that you can only do once you start becoming capable of representing future states, of simulating different circumstances and outcomes, which also means that you gain the possibility of using your past experiences to represent, to simulate a future. All of that thanks to having these associative capabilities that you get with the cortex, which then builds on and gets you even more complex capabilities, like you become capable of representing your own processes. You gain the possibility of self-awareness, but you become also capable and competent of representing the internal states of others, what you simulate the awareness of others, which makes you capable of taking others into consideration, and that is what is the basis for what I like to think of as our ultimate level of complexity in life, which is building a culture, living in society and building a culture. All of that because there's this cerebral cortex with units that are capable of putting things together and then acting back, modifying back what other structures actually make the body do. Now, why did I refer to the cortex as a saddle on top of the rest of the brain? Because that's what we've learned in recent years from a developmental look at anatomy. This is building on the work of Luis Puelles. I highly recommend the series of papers by Charles Watson making sense of all of these findings. But what you see here is a schematic of not a human, not a mammalian brain, but of an amniote brain, and quite possibly all vertebrate brains, where you have that what will be that brainstem. You have the diencephalon here. This is the axis of the neural tube. Note that the front of the brain is not the cerebral cortex. The cerebral cortex is not the most anterior part of the neural tube. Really important when you start thinking of things in terms of development and gradients, like Nenad Sestan was just doing. So the hypothalamus is not your most the end of the. The cortex is not the anterior end of the neural tube. The hypothalamus is, which is really important because the hypophysis develops right here in front of the hypothalamus. So the brain forms already in connection with the body. A whole other story there too. But my point is, this is what becomes the cortex. So you see, it's part of the second neuromere, the second segment of the upcoming brain, and it's really just this dorsal pouch. It's this dorsal cap of the second neuromere that will bulge out to different extents depending on a variety of factors. But we know that you get neurons that vary by a factor of 1,000 when you compare a mouse pallium to a human pallium. Really important thing, especially once we start focusing on the comparative aspects of cognition across species that have different brains of different sizes, different numbers of neurons, we realize is that this pallium is the same structure in birds and in mammals. The only difference is that we call it a cortex in mammals because it is very obviously layered. But you see that the part in green here, this is the entirety of the pallium in a bird brain and in a mammalian brain here. Like I mentioned, at first sight, the organization seems very different. You have these layers in mammalian structures. You don't see obvious layers. By the way, you don't even see obvious white matter, gray matter segregation in bird brains. But if you all you need to do is use a trick of light, use different light, and the different patterns of connectivity will be revealed in a bird brain, and you realize that the same organization in orthogonal patterns of fibers that you find in a rat brain, they're also there. What's so important about this is that you do have the short connections that build columns, that build functional columns, and you have the long-range connections orthogonal to them that create this possibility of associations that I just proposed to you is what makes the cortex really this structure that we focus about when we talk about flexible behavior and where it comes from. Now, of course, next part that we care so much is, you're talking about birds and mammals, but as everybody loves to remind me, aren't they connected in different ways? Isn't that what really matters, how different parts of the brain are connected? Yes, you're absolutely right. And look at that. When you look at the whole brain level of connectivity across different parts of the brain this is in a pigeon. That's the work from Onur Gunturkun's lab in Bochum a few years ago. You find that you can represent the connectivity of that pigeon brain in a way that you would not be able really to tell apart from the connectivity across areas in a monkey brain or in a human brain. And I want to focus, of course, on what maps onto the prefrontal associative areas of the human cortex, which you see here are these most densely portions of the pigeon brain. And these are areas that are organized around this main cluster here. That's the Nidopallium caudal-lateralis. I hope I said that right. We do need better nomenclature. But anyway, why bother about all this? Why am I showing you all of this? Because my next point is, of course, that given that we're talking about the same structure, whether you call it a cortex or a pallium, whether it looks layered to the naked eye or not, and this is a structure that is also organized in connectivity at this level in very much the same ways, we should care then, it should matter how many units compose these structures, the cortex as a whole and these associative areas in particular. Since what the cortex does is, by my working definition here, is add flexibility to behavior, then the best predictor of cognitive capability, the capability for flexible cognition and behavior across different species should be simply the number of paleo-neurons. I'll use that word from now on. And of course, associative neurons in particular. So let me share with you data that we published last year, again from working in collaboration with Onur's lab. what we did was compare numbers of neurons in the pallium as a whole, but also across different functional parts of the functionally different anatomically different defined functional parts of the brain. And you see that we had not the smallest brains, bird brains around, but we definitely had the biggest brain that exists in birds today, which is the ostrich brain, which is pretty respectable. It's about this big. And you see that here I have the species ranked by brain mass, but if you rank them in number of paleo-neurons, then you realize that the three crow species that we had in the sample, even though they have brains that are much smaller than the ostrich brain, they have many more neurons. The numbers I'm showing you are numbers for the whole, for the two hemispheres of the brain. And in particular, the number of nidopaleo neurons in those species is pretty large, and it definitely at least twice as large what you find in a much larger brain, which is the ostrich brain. Now, to be fair to ostriches, to the best of my knowledge, nobody has really asked them how capable they are, so we don't know exactly how capable of flexible cognition ostriches are, but we know that crows do really great. Now, what does that mean in the context of other species, mammals in particular? Well, we know that in our case, we're certainly not the species of mammals with the biggest cortex, but we are the one that has the most neurons. Gorillas and orangutans have half as many neurons as we have in the cerebral cortex, which is about 8 or 9 billion neurons in those species versus our average 16 billion neurons. And what does that matter? Is there really a correspondence between numbers of units in these structures and performance? Here you see the performance measured by the way, this is performance in two tasks that measured the ability of inhibitory control, which is a flexible cognition task. And you see the performance higher is better performance, up to 100%, so you have a ceiling effect here. But you see that for when you compare bird and primate species according to the size of their according to cranial capacity, you see that there's this very clear separation between birds and primates as if they were two completely different worlds, really. But once you just forget about the size and focus on the number of paleo neurons that you find in these species, you see that everybody lines up. You have overlap now between birds and primates. You have crows reaching ceiling level of performance here already with macaques, and as you have in great apes as well. And notice that macaws are pretty high up there with tons of neurons in their cortex. Now, what does this tell us about the human brain? So how does this compare to the human prefrontal cortex? What we found is that when you use the same method, which in short consists of dissecting the parts of the brain that you care about and then just turning them into soup so that you can very easily and directly count numbers of neurons that make them up, you realize that, and you do that again and again in a number of different species, you realize that this is us here. We're not outliers. We're not extraordinary. We do stand out in that we do have, like I told you, the most neurons in the cortex of any species, but it's still exactly what you would predict for a primate with our overall number of neurons. The same applies to the volume of the white matter as a function of the number of neurons. So this is the primate line here. I'm showing you primates in red. And if you now focus focus on just the primates and break the cortex down into associative areas, meaning the pre-callosal areas by this definition, which is the straightforward anatomical definition for doing comparisons across multiple species, you see that the line that you plot for the non-human species that relates the numbers of neurons in prefrontal cortex to non-prefrontal neurons predicts precisely where you will find the human species. And also, that is also true when you look at the volume of the white matter in those associative areas as a function of the number of neurons in that human cortex. How do these numbers compare to the numbers that I just showed you in corvids? Well, Mariana Gabi, a PhD student in my lab, found years ago that in all the primate species that we analyzed, humans included, these associative areas represent 8% of the entire number of cortical neurons. That means that humans are no exception, but the fact that we share with other primates having 8% of all of our cortical neurons be those associative neurons means that we have a lot of them. We have the most neurons in these associative areas. That's over 1 billion neurons in these precallosal areas alone compared to what you find in the other species. Whereas in birds, just because the distribution is different, in the crows in particular, we find that 40 to 50% of all the paleo neurons actually are in the I'm just going to call it NCL, sorry. About 40 to 50% of the paleo neurons are in the corvid NCL, which means that crows have 300 to 400 million neurons in the NCL and in the nidopallium, sorry, not NCL, the nidopallium as a whole, which is on par with what you find in chimpanzees. And even a pigeon, even a lowly pigeon, sorry pigeons, falls in numbers of neurons in the nidopallium between a marmoset and a monkey. So birds do pretty well, thank you, especially when they are crows. So all of this sounds wonderful. All of this sounds like benefits. You get all these extra things that you can do. Now, what about costs? Surely there must be a cost that comes with all this if it holds, even in biology as a whole, that there is no free lunch. And certainly there is a cost. So an easy way of grasping this is consider neurons your brain Legos. They're the functional units that build brains. So the more units you have, the more possibilities you have, of course, of building ever more flexible, complex, intricate patterns, right? But also, as you can imagine from how long it would take to build these different houses with just a handful, a bucket, or a truckload of Lego pieces, of course it takes much more energy and particularly loads more time to put together a system that is made of many, many more units. And that does have a very direct and I think extremely important and consequential impact on life as a whole and human life in particular, because as I published this a couple of years ago, it turns out that forget body size, the best predictor of longevity by far is simply how many neurons a species has in the pallium. And I mean species because you see this is warm-blooded species. Mammals as a whole, or there are all the colored points here, primates in red, this is us, right on the mark. And in black here, you see a number of bird species. So you see that it doesn't matter if you're a bird or a mammal, or even if you're a mammal, if you're a human, the more neurons you have in the cortex, the longer you're gonna live, and humans are no exception. Now, notice that that also comes with what is shown in the graph to the left, which is that the more neurons you have in the cortex, the longer it takes each of these species to reach sexual maturity. Why do I care about sexual maturity? I happened onto sexual maturity as a proxy for when is an animal, an indicator of when an animal is cognitively capable enough that it can reproduce and care for its young and bring them to the next generation with success. So sexual maturity is a practical indicator of age of cognitive maturity for me. And that increases directly with the number of neurons in the cortex. And let me sum all this up for you with how I like to view in one image, how I like to view vertebrate brain evolution. The number of neurons that you have in a circuit, cortex as a whole, prefrontal cortex in particular, gives you capabilities. But those are that's it. Those are your biological capabilities. It takes time to use those capabilities, and through trial and error you actually build abilities. So the more neurons that you have, the more of these capabilities that you're gonna have. It also comes with more waking hours, which gives you more time to actually do things, whole other story. But it also comes with more time to develop, so a longer childhood, more time for you to practice doing whatever it is that you can do, that you're gonna find out that you can do, and also longer to live, which means that you're gonna have more and more time to keep doing those things that you just found out that you can do. Increase those numbers even more, and you get to the point where you're so able, you've developed so many skills that you start being able of creating technology. The point of technology, I'll be happy to tell you more later, but to me, it's that it gives you free time. It allows you to use those capabilities to other ends and to do different things with what you have. Now, add even more cortical neurons that you start living so long and having such a long childhood that you have generational overlap. Put these things together and you have the possibility for cultural transmission. That is maybe this ultimate expression of complexity, of the complexity that a cortex in general and a prefrontal cortex in particular can build. And notice that you only get to keep that complexity alive by recreating it all over again in the next generation. And for that, just getting that number of neurons and the connectivity and whatnot that you get in the cortex is absolutely not enough. You have to go through all this to develop everything again and train that cortex into becoming what it actually can do in every new generation, in every new individual, which I think is a pretty useful thing to remember, that also I think makes the comparison between mice and humans not straightforward, because how much opportunity has a 2-month-old adult mouse had to develop its cognitive capabilities into abilities compared to a young adult human who has had 20 years to do that, right? Not a fair comparison. So I'm going to stop here and thank my collaborators, and thank you so much.